So my name is Paulina. I'm a computational biologist at Gladstone working on Archer, which is a package for analyzing single-cell attack-seq data. And that's actually how I got to know about Latch, because I wanted to containerize Archer so that we didn't have to deal with all the dependency hell. And then I'm doing a Shiny app, so that's also how Latch came to be useful for us. And hopefully you can use Archer on Latch pretty soon. And today I will be presenting Enformer. So I hope you have heard about that. Let me see if I can share my screen. Can you guys see it all right? Awesome. Okay, so Enformer is a paper that I have been very excited about, just because I feel like a lot of the tricks they did were very creative, and the future applications of this are very promising. The outline for my presentation today, I hope to first go through the background and motivations, then the model architecture and benchmarks, and then the applications in biology data sets, which I think was the bulk of the paper. But because of that, that's why I'm leaving it towards the end. And then hopefully discuss with you some key takeaways. So for any of you who are more on the software engineering side, I want to give a little bit of biology background. So when we talk about regulatory elements, some of the ones that come to mind are promoters, enhancers, and insulators. So promoters, an important thing to know about them is that they're usually at a predictable location immediately upstream of the transcription start site, whereas enhancers, they're not as predictable, like where they will be. Sometimes they're very far away, and they act through 3D interactions. They also help to determine cell type specificity. So they're of interest for people studying like developmental biology. And the third type of regulatory elements that is relevant for this paper are insulators, specifically topologically associated domain regions, which help to minimize enhancer-promoter crosstalk. So I'm not sure if I have an example here, but usually they can help upregulate or downregulate how a gene is being expressed. And so when we talk about sequence-based models, there have been many before Enformer, actually. And so Enformer is trying to build and improve upon them. If you've heard about Avsec, Akita has to do more with the 3D genome. Basenji is the one that is talked about most on the paper and sort of used for benchmarking Enformer, specifically Basenji 2. And then BPNet is one from the Kundaje lab that Ziga, the first author, also worked on, but I don't think it's mentioned in the paper. And if you haven't made the relationship, all of these, or most of these names come from cats and dogs. So the motivation for the paper, also feel free to interrupt me, by the way, if you have any questions, because I'm not looking at the chat. So the first motivation was translational. They wanted to help with a field effort for fine mapping. So fine mapping refers to disentangling causality from association due to linkage disequilibrium. And I mean, I think there are 1-hour talks on what linkage disequilibrium is, but really, if you think about it just from a statistics point of view, it's identifying, like, attributing the causal effect to one specific single nucleotide polymorphism. And because there are many significant ones, knowing which specifically are the causal variants. The second motivation was to increase information flow between distal elements. Here on the lower part of the screen, you can see this is a figure from Basendi 2. And the problem with deep CNNs and architectures like the one from Basendi 2 is that they have a very narrow receptive field, so they cannot capture regulatory elements like enhancers that are influencing the gene expression from long distances. So they're only able to consider nearby sequence elements. So I looked this up on Google, like how can we increase the receptive field in a CNN? Because I'm sure it was one of the first questions that the authors were thinking of when they were trying to improve Basendi 2. One of the options is to add more convolutional layers, like make the network deeper. Obviously, this has some hardware implications, and so it's not necessarily feasible. The second one is to add pooling layers or higher stride convolutions. The third is to use dilated convolutions, which is actually what Basendi 2 used. And the fourth one is to use depth-wise convolutions. And I'm sure you can do like a combination of these, but these are some of the answers that came to mind. And then another option, which is actually what Enformer does, is to use transformers. So Enformer is, as you may know, not a name of a cat and dog, but actually a portmanteau of enhancer and transformer. It has three main parts. The first one is 7 convolutional blocks with attention pooling. The second one is 11 transformer blocks. And the third is a cropping layer followed by final pointwise convolutions, which branch into 2 organism-specific heads. Is someone trying to speak?
I think a bunch of people just joined.
Okay.
Unmuted.
No worries. So here we have human and mouse, and if you noticed, we're predicting more human tracks than mouse, and that's just because we had more human data. Okay, so like I said, I wanted to focus and start with the model architecture because the biology applications are the focus of the paper. So the input is 1 hot encoded DNA sequences, as you can see here with the dimensions. The left side of the blocks here, and this is all in the supplementary information, which is kind of sad because I feel like I was kind of hoping that they would talk about this mostly in the paper, but in the left side of the blocks, they have the number of trainable parameters. Interesting, so they start increasing with each block, and then after the pointwise convolutions decrease to 4 million trainable parameters. Then on the right side of the blocks, you can see the output shapes, like I mentioned, starting with the 1 hot encoded sequence, and then going on to, so they go from, let's see, so from half the number of channels to the number of channels that they wanted, starting from a fraction of them, as well as, if you can see, the same thing is done in Basendi 2. So it's only, they start with a fraction of the number of channels and then reach to the total number of channels. And I will talk about why this is like common practice or a good idea in the next slide. So the 2 main hyperparameters of the model are the number of transformer dilated layers, L, so as you can see here, and the number of channels. And I think one interesting thing to note here is that they say that at the end, at the end of the transformer, the number of channels is 1536, but here also the first dimension is 1536, so it's actually the output shape after the transformer is 1536 by 1536, and I don't think there's like a reasonable talking to like friends and just being curious about why this is, like, you have any idea of what would be the rationale behind this? I'd be interested in hearing it. So like I mentioned, they start with a fraction of the channels and then increase. And this is something that I wasn't too familiar about. Why is it necessary to increase the feature channels as the network goes to deeper layers? So the first reason is that deeper layers have larger receptive fields and generally correspond to more specific or complex features. So you want to have more channels to capture them all, which makes sense, right? And then the second one is to apply pooling, that as you apply pooling, your data gets smaller, and that way you can afford to have more channels, keeping the same amount of information, but making it more relevant to your problem. And that was just like a theoretical question that I had. Now, moving on to after the input, the two first blocks are the stem and the convolutional tower, and together these form the 7 convolutional blocks. They help to reduce the spatial dimension from 197K base pairs to 1,536 base pairs, like you see here in the output size, so that each sequence position vector represents 128 base pairs. So that's sort of like the effective receptive field that they're working with. And then attention pooling is one of the main innovations in this paper. It helps to summarize a contiguous chunk of the original input sequence. And I'm sorry for the math here, but I think this equation is not a commonly used one. Some people have told me that it looks similar to softmax. It's not softmax. This has to do with attention pooling. So it's something that actually they have used, and I haven't seen it anywhere else. So if you have, I'd love to check out where else it has been used. But what happens is that so in the pooling window is weighted by the exponentiated dot product of the value and the weight, where the weight is a matrix of which is weighted. So they have the pooling window, which is a matrix of the learned weights, and that's sort of like the attention filter. And something else that's important here, let me see. R here denotes the relative positional encoding, which is what I will talk about next, and is another one of the main innovations of the paper. So usually, and if you're not familiar with positional encoding, it's very commonly, you either have absolute positional encoding or relative positional encoding, but it's sort of a main part of the transformers. And positional encodings are embeddings that have both meaning and position of a word, and they're needed because input embeddings aren't fed sequentially like they would be in an LSTM. And so you need to keep that kind of like positional information, as the name suggests, and feed that into the model. So it's usually popular to use wave frequencies to capture positional information. That means sine and cosine. And this comes from the original Attention is All You Need paper. So this is not something that the authors came up with, but very creatively, they added more functions in order to more easily distinguish positions upstream and downstream of the transcription start site. So this is like something that SIGA came up with, according to the podcast that I heard when he was explaining like how they thought about adding more positional or more functions and information to the positional encoding additional to sine and cosine. He said that initially they had just used sine and cosine like people in the field traditionally do, but then they performed just as well as BASEN-G2. So the motivation behind adding these functions was giving the model a sense of the direction of the regulatory element. And so I'm not going to go through like y exponential, y gamma, and y central mass, but I think it's very creative, and I honestly don't understand like the math behind it.
I have a
Um,
question here, actually, Paulina. Um,
yeah.
yeah, so for this, I mean, it seems like nothing here is specific to the biology or the encoding of the input as nucleotides, and that having a richer understanding of both sides of your single positional character is generally useful in NLP. Have you seen people like take this result as it was developed for the gene motif prediction and apply it in just biology agnostic domain?
I have not, but hopefully they have, because I'm sure it's very useful, like you're saying.
And I was also going to ask, you might have just said you don't know. It seems like the gamma is a bit of black magic. If the authors discussed like the motivation behind that particular function.
They didn't, but I was trying to look at like why sine and cosine, and it's very, very interesting because it has to do with sort of like least squared bits, or how much information is encoded in odd and even pairs of these tokenizations. So I'm going to go back to these papers and trying to understand the base functions and then see why they added these other ones.
Gotcha. The sine and cosine is developed in the attention enlightenment paper, and the gamma piece is new. So I guess yeah, understanding all of it would be good.
It has some interesting interpretations relative to Fourier analysis as well.
With the gamma functions, or just the original sine cosine bit?
Just the selection of harmonics for the.
Oh, interesting. Okay. Yeah, Stanley, if you want to talk about that now or later, that would be great.
Oh, you
Math.
were doing too good. This is the most fun I've had all week. Thank you. Please continue. Forgive me.
Okay, no. Okay, so the positional encodings are something you need before you enter the multi-head attention block. And we have this question about how do we tokenize, right? Because tokenization comes from the natural language processing field, and it's usually done directly on the input sequence, tokenized into words and embedded into a richer embedding space. So I feel like because language is something that we're so accustomed to, that notion of tokenizing words comes more easily than like, what is a token in a series of contiguous nucleotide letters, right? And so actually, this is a minor note that I saw in the supplementary information, but I think it's very important, is that they use the convolutional tower that I showed previously to compute this embedding of nucleotide segments and contribute an inductive bias for what a motif would look like. I mean, that's a packed sentence, but I think there's so much debate about whether we should even tokenize or how we should do it when we're applying transformers to genetic sequences. So definitely is something that I will continue looking into. And one important thing about the MHA block is that they use 8 heads. One head outputs filtered value matrix that assigns high focus to the features that are more important. This obviously contributes to the attention side of things. And you need many heads because each has different attention filters that will zoom in on different elements. And each head does that by having a separate set of weights. So not only does each head has different attention filters, but within each head, you have weights for the queries, the keys, and the values. And those are the ones that transform the input sequence into the attention filter that you need. And then if you see I think it was figure 3, they actually are able to take out the attention filter and output like a heat map showing where the model is attending to for the TAD insulator regions that I mentioned in the beginning, which I thought was very interesting because it's not something you can do with any model, right? Like that sort of adds more interpretability if you can take out that attention filter. And another point here is that MHA has quadratic complexity. So even though this has like a good resolution, it's very hard to bring down the model to a base pair resolution, like for example, BPNet, because of this quadratic complexity and our hardware limitations right now. The last part of the model, and I don't know how I'm doing on time, hopefully okay, is the cropping layer and the pointwise convolution. So they crop 320 on each side to avoid computing the loss on the far ends, because if you think about it, if your sequence, you're at the center and you're 320 away, then just looking at one side, you will get only towards the center and not past it. So it's like biasing the model in one way that we don't want it to. It won't have like that far end receptive field that is giving it its performance gain. And after that, you just have the two output heads that I mentioned. Let me see if there's something else here. No. Okay, so to summarize, the main innovations compared to Basenji 2 is that the input is 1.5 times longer of a sequence. So Enformer has 197 KB instead of 131 KB from Basenji 2. The second innovation is, of course, that they use transformer blocks instead of dilated convolutions. The third is that attention pooling that I mentioned, and that's why I think it's so important to go through the actual math of how they came up with this function and what's the difference between that function, max pooling and average pooling. And they talk about how they initialize the weights. So that's all in the supplementary information if you're interested in looking more at it. And then 4 is that they use twice as many channels. Okay. So some of the limitations are that the model can only predict for cell types and assays in the training data and cannot generalize to new cell types or assays. And then I mentioned with the quadratic complexity that training deep self-attention at scale is a memory challenge. And even if they were using TPU and Sonnet, which is from DeepMind, is like the Keras version of DeepMind, I think that's how they describe it, they cannot get it down to base pair resolution because that would require a very much longer sequence. And further research directions, if any of you are interested in working on this model, is to increase the resolution and quality of the target tracks, to curate data from additional organisms. They mentioned combining it with 3D DNA contact model like Akita. So it would be more like an end-to-end model and more comprehensive. And the last one is to improve the sensitivity of the model by training on more functional genomic data sets like CRISPR perturbations and MPRAs. So they mentioned that even though they use these only for evaluation because the data sets were too small, I think this paper was released like 2 years ago, and the amount of like the pace that these data sets have been growing is incredible. So I'm sure there will be more now. And then when we talk about applications, and it's something, some examples that they gave in the paper is fine map to existing GWAS studies, prioritize rare or de novo variants observed for rare disorders, and impute regulatory activity across species to study cis-regulatory evolution. Now, that was all of that was like the model architecture section. I can talk about the experimental results, but I'm not sure if you want to go into discussion or not. I think the experimental results are also super interesting, and it's what they spend the bulk of the paper on, like I mentioned. What do you want to do, Kenny?
We have like 14 minutes for presentation, so feel free. I also think it'll be really interesting.
Awesome. Okay, so the 4 main experimental results, starting with improving gene expression prediction. First, I'm going to give a bit of background because they use a lot of terms here. So CAGE is cap analysis gene expression, and it's just 5' capture of mRNA that's used for training the model. SHIP-STIC is chromatin immunoprecipitation of histones or transcription factors, and then DNAs, and this should be a slash or a TAC-seq, measures accessible chromatin. If you're not familiar with these genome tracks, these are peaks that are called for regions of DNA that are enriched in the assay. So the first experimental result, here you can see on the left, Enformer compared to Basenji. And what they're doing is comparing across various cell types and tissues how Basenji performs, Basenji 2 specifically to Enformer. And this is CAGE. So I mentioned in the previous slide what CAGE was: CHIP histone, CHIP transcription factor, DNAs, and a TAC-seq. You can see that for all of them, the way I like to say it is that it's to the left of the diagonal. So it's Enformer has more predictive accuracy, and gene expression predictions are also better captured for tissue or cell type specificity. So here they're doing across CAGE experiments and specifically focus here on variants. So the variants across genes. For me, this is not like, I don't see as strong as a result on the data like leaning towards Enformer, but I can see that most of it tends towards the left side of and left upper side of the diagram. So okay, so let me see. Now, this second experimental experiment they did was very interesting because this is where I mentioned that they took out also the attention filter and really wanted to interpret what is Enformer attending to. To do this, they computed contribution scores for several genes with CRISPR-I validated enhancers. And what contribution scores help us understand is what part of the input sequence are most predictive for the expression of a particular gene. And the way they computed contribution scores is through input gradients, which apparently is very common. And it's not only specific to transformer models, that's by taking out the gradient and the input. And this is tissue or cell type specific because input gradients are computed with respect to a particular output CAGE sample. Then the second way of computing the contribution scores is actually only possible through models that use attention. And they rely on taking out the weights that are internal to the model and shared among all tissue and cell type specific predictions. The third is in silico mutagenesis, which is the same as input gradients in that it's tissue or cell type specific. The results is that they found Enformer contribution scores prioritized validated enhancer gene pairs with higher accuracy than Basenji 2. And they say in the paper, quote, the contribution scores of Basenji 2 were zeros for sequences beyond 20 kb from the transcription start site. I cannot see that in their figure. Like Basenji is orange and I cannot, I mean, it's not easy to see, but what you can see is that compared Enformer to Basenji, in where it has the distance, like shorter distances, it performs pretty similar. And then towards longer distances, which is what the authors are claiming are the strengths of the model, that's where Enformer performs better than Basenji. So why is this remarkable? Enformer uses only DNA sequence as input and was never trained to explicitly locate enhancers. And more surprisingly, Enformer lacks experimental data. whereas models like ABC, which you can see perform here, it's performing worse, but here better, like all the green versus the blue. ABC actually takes as input experimental data. So Enformer is performing sometimes even better than it. Okay, third experimental result, improving variant effect prediction on expression quantitative trait loci. So here they looked at data from the Genotype Tissue Expression Project, and their challenge, and usually the challenge when doing this kinds of predictive validations, is that you have a rippling effect of causal EQTLs to nearby occurring genes due to linkage disequilibrium, which is that phenomenon which I referred to when I was presenting fine mapping. And one technique that has been used to sort of get around the linkage disequilibrium noise is to measure the genome-wide statistical concordance between signed variant annotations, such as the model predictions, and GWAS summary statistics, such as the ones that are provided by the GTEx project. And this accounts for linkage disequilibrium. And so, as you may predict, the Enformer is able to improve variant effect prediction with a higher C-scores than Basenji 2. And then here, this is just on the lower side of the figure, is just showing that Enformer had a statistical significant difference in results in the AU-PRC compared to Basenji 2 when it tried to identify specifically causal variants. And actually, this is where they used the random forest classifiers, which I think was just like, they didn't have to add this to the paper, but I like, if they wanted to do more work, sure. And to predict this causal variance, they derived features from Enformer and from Basenji 2, and they found that the features from Enformer were more predictive than the ones from Basenji 2. And then this is my final slide. It's very packed, I'm sorry, but it's the final result. Enformer improves non-coding variant effect prediction as measured by saturation mutagenesis experiments. And what you can take out from this lovely heat map here is that everything above the horizontal break, so this white line here, is methods that require no additional fine-tuning on each locus. So from the methods that require no additional fine-tuning, Enformer performs the best. And when it's fine-tuned to the data of MPRA experiment used for this validation, Enformer with last regression had the best average correlation across all loci of the promoter and enhancer pairs. That's my presentation.
Awesome job. Really great ability to distill complex concepts in this